to be consistent.
I'm asking, like, are we still going to post on your channel?
No, we can post in your channel.
Okay, I'll make a channel then. Okay, so hi everybody, this is March 30th, 2021. This is our first general body meeting for Drexel AI. First day of the spring term, and I just want to welcome everybody. My name's Ethan. I am an undergraduate here at Drexel studying biomedical engineering with concentration in biomedical informatics, and I have a very deep interest in Drexel AI. I started here back in fall term this year. We worked on two very interesting projects on neural network architecture search and in fall, and then we did two projects on stock market predictions last term. It's been kind of a cool experience. So we can go around and introduce themselves. So Yit, do you wanna say something?
Yeah, sure. Welcome everyone. My name is Yit. I am studying computer science and computer engineering with BSMS program. I am hopefully graduating this term. I started this club about 2 years ago, and we had many projects. We participated at many hackathons. We started very small, but we grew a lot, and the starting point for us was to help dragons basically get familiar with these fields before they start to take these ML AI classes, so that they can talk about them during their co-op interviews, maybe in their applications and so on. So we put a very strong emphasis on projects and papers. And we held elections last term. That's why I'm no longer president. And we are also planning a very big conference this May that we are very excited about. And I hope to see everyone at least present something. So when you're taking a class this term, make sure that you do a project and write a paper in a conference style and then submit that to the conference, to our conference, so that you can present.
Awesome. Alicia is the... Yeah, she's here. I can't see her. Yeah, she's the vice president. Would you like to say some words? And we can just go around and introduce everybody.
Hello, everyone. I'm Alicia. I'm a second-year undergrad computer engineering major with minors in mathematics and neuroscience. I do have a strong interest in AI, machine learning. I've been a part of Drexel AI since last year, my freshman year, so fall 2019, and now I'm the incoming vice president.
You can just call on like somebody else. We can just keep popcorning around.
Jeff, do you want to introduce yourself?
Sure. So my name's Jeff. I guess my title is co-secretary. But yeah, I'm helping manage the conference that we're hosting. I'm a senior in undergrad doing a dual degree in math and computer science. Yeah, I work in the same lab as Yeet and Isamu with Dr. Kim. I do like, machine learning stuff. So. Oh, and then I should pick somebody else, I guess. How about Adam?
Hey, thanks, Jeff. I'm Adam. I'm a third-year undergrad mathematics major with a minor in computer science. Right now, I'm doing my co-op with a company in New Zealand to try to use AI to solve the housing crisis. And I am the events coordinator for Drexel AI. Let's see. Who hasn't spoken yet? Isamu, have you spoken yet?
No, not yet. And Yeah, hello, my name is Samuel. It's been quite a while since I last came to one of these meetings, but I'm quite interested in like reinforcement learning and doing machine learning projects. And yeah, I'm in the same lab as Jeff and Igit, and I always love your cat, Jeff. And for the next one, maybe Dan, can you go?
Hey everybody, my name is Dan. I'm a finance and accounting major. I actually just transferred in Drexel in the fall. So unfortunately due to COVID, didn't really get a chance to connect too much, and just finished co-op where they were doing a lot more coding, not necessarily AI, but just kind of picked up on a lot of that stuff during my time there, and looking forward to kind of diving deeper and just learning more about everything AI related in general. Go. That's all. Pick Molly.
Hey everyone, it's Molly. I'm a second year in electrical engineering at Drexel. I previously worked with Dr. Das on reconstruction of visual experiences by using convolutional neural networks. And currently I'm on a co-op working at Dr. Rosen's lab in bioinformatics. Mainly we're doing like human microbiome analysis using machine learning techniques. Is there anyone who did not talk?
Sesh, Abe, and two others, right?
Hugh and Palash. I don't think they spoke today.
I can go if you want.
Sure.
Hi, my name is Shesh. I'm a chemical engineering major. I'm interested in machine learning and reinforcement learning. I joined Drexel AI in fall of 2020. And yeah, I just looking forward to this semester or this term.
I'm Palash. I'm a data science major, computer science minor. I just finished my co-op at Dr. Rosen's lab. I think just picking, I think Miley is picking up where I left off. And
what did you work on at Rosen's
I
lab?
was working on I think we are working on slightly different projects. I was working on collab with Fox Chase. So I was working on cancer, like mutational variation analysis, basically trying to identify variants that are correlated with certain kinds of cancers.
Okay.
But it is the same lab, so I was kind of,
Yeah, cool.
I didn't meet you. Yeah, but yeah, excited to join the group. It's my first meeting.
Is
You're welcome.
Joe still working there in Dr. Rosen's lab, or did he graduate? I was working there when I was, if I remember correctly. Okay.
Call on somebody else. Abe, would you like to go?
Yeah, sure. So, hi, my name is Abe, and I'm a first year computer science major. So, I don't really have much experience with AI, but I think I'm pretty interested in learning more. So it's great to be here.
Awesome, welcome. And then I think we have one more person, and I'd hate to butcher the name. Could someone help me out? I'm serious.
You mean me?
Yeah, you. How do you pronounce it?
You can call me Hugh.
Hugh? Okay, thank you so much.
No problem. So my name is Hugh. I'm currently fourth year at Drexel University. I very interested in AI machine learning when I first choose my major, and currently I took AI track right now. With my first class is artificial intelligence with Professor Popyak. I don't know. I don't know how to pronounce his name.
Cool. Well, welcome. So I think it's pretty interesting that we have a good bioinformatics basis. I'm pretty bioinformatics heavy myself, and it's good to see that there's other people in the lab other people in the club who have interest in that as well. So last term and the term before, we focused mainly on constructing these projects, and we took some of the projects to paper publication, to conferences. We spoke at the Harvard conference that was in the fall or the winter, and then we submitted 2 other papers to IJCNN, which will be occurring in the summer through IEEE, and I think we're still waiting to hear back on that. I think we get results back in April, so that's pretty exciting. So I think that it'd be interesting to kind of discuss what kind of projects we'd want to consider for this term, for the spring term, and what kind of research areas we're interested in. I know in past meetings he had this elegant first meeting slide where he kind of explained what machine learning is, but I do not have that. So is there anything, I heard a lot of reinforcement learning, and that's actually a topic we talked about a lot in the beginning weeks of winter term, and we kind of discussed how there really is only two kind of extremes for implementing reinforcement learning that we know of. On the one extreme, it's typically video games and like Flappy Bird, Geometry Dash, but on the other extreme, there's more complex problems, and which we explored was stock market prediction. And we could continue with that project, but we kind of reached at a standstill right yet, where it really didn't move anywhere, at least with our current implementation. So is there any other topics that people are interested in per se? My background is kind of in a little bit of neural nets, and I've kind of implemented like naive Bayes, random forest, decision trees, KNN. I'm trying to think what else. Those type of like supervised methods, but I guess that we really have to formulate a problem before we deep dive into choosing a method. I mean, the discussion is open.
Yeah, I mean, I've done like the stuff that I've worked on in the lab that I'm a part of, along with like Osamu and Yeet, is I guess classified under like representation learning. So we could be doing something like that. I don't know, like sparse coding.
Could you explain what representation learning?
Yeah, Yeet, correct me if I butcher this, but it's like you're extracting. You have like some data and you want to describe it in terms of a collection of features. So like problems that you deal with are like feature extraction under data, and then like, dimensionality reduction. So like maybe if you have a picture of different types of fruits, you could have a feature for each fruit. Like that's a big like holistic view, but like, it gets more complicated. Like you can have tiny features that are like edges or corners or something, but you can also have like, holistic features like this is a dog or this is a cat. I mean,
okay. How similar or dissimilar is it to like feature mining or feature extraction? Or is that just one component of it?
I mean, I think feature extraction is like part of the problem that you work on. Like you want to identify features that are important to like representing a given data set, or or an image, for instance. Like, and then like your representation is like a set of coefficients that you map onto the set of features that you've identified, if that makes sense.
Yeah, okay. That's interesting. So right now we got representation learning, something we could explore. It's something we'd possibly want to put in under our tool belt. I think maybe a couple times last year, we actually had some people present on topics that they individually looked at. I think there was one presentation on reinforcement learning, and then my friend Will came in to talk to us about how he actually how he trades, does day trading on the stock market, and that was particularly important for when we wanted to explore pattern prediction last term. Any other thoughts with types of either methods or an interesting problem? I've always been a proponent of problems in bioinformatics. I explored something known as the protein folding problem last summer, feels like ages ago. It's the idea of, given amino acid sequence, how do you best predict its three-dimensional structure? It's since then been very explored by Google, and they've kind of hit Eureka, hit a pot of gold with what they've been able to do in the past couple months. It's been pretty interesting.
It seems like we are strong on bioinformatics this
Yeah.
term. We could push a paper.
Yeah, we could push a bunch.
Combine that with representational learning that Jeff mentioned.
Yeah, I mean, the stuff that we've done is heavily like neuroscience influenced. So like, I mean, even that is sort of, I guess, bioinformatic type of.
Yeah. So I actually had a thought the other day about kind of an unsolved problem. It might be interesting to talk about it. It's very bioinformatics based. So the idea is that, so biology, there's this mantra that there's this mantra that like structure dictates function, right? So if you know the structure of a protein, you basically know what it does, and you can very well categorize its function, how it plays in a pathway, or how it exists in a membrane. You know, whether it's hydrophobic, hydrophilic. Very biointensive for those uninterested. But what's not obvious is the opposite, right? You can't really take a function and then build a structure, and that's actually the most useful thing we want to do, right? We want to be able to say, okay, I want a protein that does this, and let's build it. That would be, re-engineering a protein, which I think would be a really interesting problem to look into, but I would have no idea how to go about doing it. One thing there is that we can look at, if we'd be interested in looking at this, is something known as structural motifs. So there are like sequence of motifs in DNA that are reoccurring across the genome, and then there's structural motifs that are reoccurring across protein structures. And so those would be important in, like, developing an enzyme with an allosteric site, or developing an enzyme that cuts at a certain point along a gene, or creating an enzyme or ion channel that allows particles to flow through a membrane. That to my knowledge, that the problem of going from function to structure is kind of unexplored at this moment. Sure.
Yeah, so these like structural motifs you're talking about are like patterns you see in specific proteins that like have specific types of functions, but there hasn't been much work to like correlate the two, right? I guess.
Yeah, to put them together. Ideally, you'd want to say, ideally you'd want to like do a Google search, and you do a Google search for some like function. You'd say, I want a protein that cuts DNA at this site, and it would spit back the amino acid sequence, and you'd effectively know what DNA sequence to synthesize to get that protein. That would be the end all. Obviously, we're far from that, but I think trying to use, trying to like apply machine learning onto that topic would be pretty interesting. It'd be interesting to just kind of formalize how you'd go about doing that. But yeah.
I have actually many questions about it. Is it one-to-one between structure and function? My other question would be, how does a structured data set look like? Is it XYZ coordinates on floating point numbers, or is it just an image?
Yeah, so
data set.
When I was dealing with protein structure, what I did is I had basically a 3D object, where a 3D array of objects, where each object kind of stored features about the protein. So if you could imagine that there's one feature in the grid of objects that has like the occupancy of the nearest atom. There is another feature that kind of stores the like the name of the nearest atom. So whether it's like a carbon, nitrogen, phosphorus, sulfur. There's another feature which dictates the position of that atom in the structure. So whether it's like an alpha carbon or a beta carbon, those are carbons in the like an amino acid in amino acids in an amino acid. And back to your other question, no, it's not one-to-one, right? You can have it's like it's the same idea with programming, right? You can have you can want to make an array that goes from like 1 to 100, every like only even numbers. You could do that many different ways in Python, many different ways in C, many different ways in Java. So that's kind of analogous to the problem we're kind of facing here. Like there is the best case scenario, but there are other plausible scenarios that you could implement or that you could use for a protein to do some sort of function. I do have a library built already in Python that encodes proteins into those three-dimensional structures that a machine learning model can recognize. And I did explore different metrics that would attempt to measure how well protein folds. So that might be relevant here if that is a route we wanted to go down.
I have a question. Do you have a database for like what this protein does? Like
yeah. So it's kind of surprising how well documented these proteins are online. If you Google like protein data bank, you search by a 4-letter ID, which is the protein, which corresponds to the protein structure. And there is a very, very well documented, I'll pull it up real quick. There's a very well documented information about protein data bank, about its function. As for like structural motifs, I'm not sure. So we Google like protein data bank. It might be here. It's definitely not that. PDB homepage, I'm hoping it's this. Yeah, it's this right here. Okay. And then I think one is ICRN. This is a particular structure, and it is a hydrophobic protein. And there are links to papers, there's literature. So the data is there, in my opinion, to kind of examine structure and tie it to function and try to do this like backwards mapping. But you need something kind of novel in the realm of machine learning to do that, or it doesn't necessarily have to be a complex model, but I kind of have a feeling that it's going to be.
Yeah, for me, I was thinking like, how can you represent the function in like a machine learnable way, and also like the protein.
Okay.
Yeah.
Can you look at proteins like this and like identify whether they're hydrophobic or hydrophilic just based on like what they look like? Is that like something like that is obvious to you, or is it just like, is it just something that's like written up in the database?
So yeah, the hydrophilic versus hydrophobic, that has to do with what is on the outside of a protein structure. So if you look at an amino acid sequence, it's not obvious if that protein is, in what regions is that protein hydrophobic and hydrophilic. In most protein, in like almost all proteins, you're going to have regions that are hydrophobic and hydrophilic. The hydrophobic ends tend to kind of fold into each other. And that's how the protein, like that's something that drives the protein folding mainly, among other kind of interactions. And then the hydrophilic parts are pointed outwards. But you really wouldn't understand that until you get like an optimal protein folding. Luckily, I don't think we'd have to do we wouldn't necessarily have to do a protein folding. What we would have to do is we would have to, like Osamu mentioned, figure out a way to kind of map function to some machine, like some learnable variable or a set of learnable features that we could kind of associate to functions of proteins, structures of proteins.
do we have to learn it? Is it something learnable, or are we just searching for it? Or
Well,
what
it
is a
depends
search
what.
space? Is it, for example,
So if we, so the most naive approach would be to search databases and form a tie between structure that already exists in downloadable files and like its annotation, right? But what I had in mind was to use structural motifs that are pre-existing that you'd use to actually build a protein. So instead of instead of like doing if you want to look at Legos, right? Instead of like doing a search between like a Lego, all available Lego houses, and then the title of the Lego house, instead of like trying to learn the relationship there, you're trying to take like smaller pieces of the Lego house and form a tie between like the structure of that piece of the Lego house. Like this is a wall, this is what the wall does, as opposed to this is a police station, this is what the police station does. So like I think the more like if the search space would be a lot different if we were forming a tie or forming a relationship between the database and functions of already constructed proteins, because the idea at the end of this isn't to simply just do a search for function to protein structure. It's to create novel proteins that have specific function.
Got it. I ask you because if you remember our search papers, the one with the architecture size, we saw that there were many architectures that would help us achieve the problem, but instead of looking at each architecture, model architecture one by one, we just did a binary search to eliminate half of the search space. So that was not something that we learned. It was just something that we kind of took from the existing literature and applied to a new domain. So I was wondering if it's something similar, but looks like it is not. I mean,
so I think that binary search, sorry.
I was going to say, so you made the analogy of having a wall for a house and a police station. Are these motifs being shared
The structural
by
motifs,
different structures?
Yeah, the structural motifs would be common amongst proteins.
Got it. So then how would you differentiate a wall in a house and wall in a police station? We have to look at the higher dimensionality. So I don't know how to do that with like structures.
Yeah, this sounds kind of like a generative adversarial network might be helpful, which is like,
actually, yeah,
like
I could see that working actually.
You try to build a discriminator that like identifies whether a given protein has a structural motif, and then you have like a separate generative network that's trying to generate new proteins that also have that same motif and try to fool like the discriminator. So it's like
yeah, and at the first pass, right, Asami was talking about how to classify or how to basically make the label something that can be like learnable or well defined, and we can just start from whether a protein cuts or not, right? And we can design we could try to find that protein that cuts or not. That is actually the perfect way to implement that. Thank you, Jeff.
So I guess the first step is to just gather data, so like web scraping or something like that.
Yeah. So when I actually, I don't know, because whenever I downloaded data from online from Protein Data Bank, I would just query using their API to get the structure in coordinate form. So it was simply just a list of XYZ for each atom in the protein. And then I would just load it into that matrix that I have. It would load it into a protein, into the protein object matrix, the three-dimensional matrix. I don't know whether we'd have to do web scraping. Maybe we'd have to have some annotations for structural motifs that we'd want to find.
Yeah, that's like, I think that would be necessary if we were going to build some sort of discriminator.
Yeah. So yeah, before we go on with like specifics, does anybody have any questions about where we are, or about the problem?
Anything is, basic stuff about like what a discriminator is, for instance, or like what a
Yeah.
generator is.
I think in terms of bringing everyone to the same page, we might need to have a session to explain what a GAN is. And
Yeah.
also, I don't have, for example, the bio knowledge
Okay.
for going deeper. So maybe if you want to have another session for that,
Yeah.
it might be
So I
helpful.
think that's a good idea. So next week, Jeff, did you cover GANs in your CS class?
Yeah, it was in my deep learning class.
Okay.
I could actually, I could do a little presentation on it if you want.
Okay. And I really should practice for my I have a conference later this week, not later this week, later this month, and it's on the protein project, so I can just present that as well next week as to give a bit of the background, and I can talk more into what features I selected for the protein. And I think we can actually, if it works how I think it's going to work, we can actually just reuse that framework for encoding proteins, which would it just takes a lot of work out of it, because that took me nearly, I don't know, five weeks in the summer to figure out. So
Yeah, that sounds like a good plan. In the meantime, do you want to create a GitHub repo and maybe put a few links to useful papers so that everyone can read?
Yeah. So yeah, that's a good point. Last term, we structured everything in terms of GitHub, and I'm not sure if everyone like used GitHub before. I'll put it on the Drexel one. I'll see if I can sign into that one. Does anybody have a cool name yet?
Pro team.
Just protein? Okay.
Yeah, you can say protein nets or something.
Protein nets.
And we can change it later.
Yeah, we can change it.
Just out of curiosity, how did you encode the proteins?
Yeah, so if you can imagine like a 3D object structure, where each object in the structure corresponds to a 1 by 1 by 1 angstrom cube in the 3D structure of the protein. I basically put features in that would best describe that space where the protein occupies. So like I mentioned, one feature was the occupancy of the nearest atom, the positional atom type of the nearest atom, the like what element it is of like carbon, nitrogen, oxygen. And those were the three main features I used.
Oh, so is it like a, well, it's not a time, but is it like a time series? Like a bunch of stuff connected to each other?
Actually, there were no pointers between the atoms. That's the interesting point. I didn't have any bonds. It was all it was only based on position and atom identity, like whether it was a carbon, alpha carbon, beta carbon, nitrogen. So I'll actually post this one. This is the presentation from a other conference I went to. This is the measuring protein structure. And this is something I'm going to end up spelling so many things wrong in doing this, but this is the presentation I gave, I think in the fall term at the Mid-Atlantic Bioinformatics Conference, which describes the project I did. Implemented a convolution neural network according to these features, and I was able to predict protein structures. This was the best case scenario, and this is the worst case scenario. And then I was able to show that the metrics I was trying to learn were at the very least learned. That was kind of the point of the project. I can't really, it would be too difficult to compare it to the gold standard. That's kind of what my professor and I thought of.
So you encode it into an image or something? Because
Not an
it's,
image, like a three-dimensional object structure.
Oh, okay. Gotcha.
Yeah, like right here, a 70 by 70 by 70 angstrom grid where each 1 by 1 by 1 cube is 1 by 1 by 1 angstroms. Yeah. So I guess the current plan. Let's do plan for. I wonder what the day is going to be. Does anybody know off the top of their head what next Monday? The meeting is going to be on Monday next week, and we're going to pick a time.
I think that's the 5th, April 5th.
All right, thank you. Yeah, April 5th. We're going to introduce GANs. That's going to be by Jeff. Introduce protein structure, encoding, prediction, and possibly mapping. That's all we have for the agenda. Does anybody have any other thoughts for what we might want to talk about? I know those are pretty two big topics.
And what about the data set? So you said you had to call APIs to get the coordinates into your script?
Yeah. I call the API using protein structure IDs. So I looked up earlier on Protein Data Bank like 1CRN, and I just know that is a protein structure.
How many proteins did you look at? How many protein IDs did you have?
Sorry, 1 second.
Or actually, how do you determine which protein to look at?
Yeah, so here's kind of what I did. I did kind of two tests. The one test was 100 decoy proteins that are 5 amino acids long, and then 1,000 decoy proteins that are 10 amino acids long. And then I did 1,000 native structures. And decoy and native is a term that means most optimally folded protein, and then decoy is a term that means not the most optimally folded protein. I picked these native structures based on whether or not, based on the percentage of atoms that were characterized as having an alpha helix or being contained in an alpha helix. So you can look at protein structures on various scales, right? You have the primary structure, which is just the amino acid sequence. Then you have the secondary structure, which can be classified as like a turn, a helix, or a beta plated sheet. Like if I go to like protein secondary structure, it should be like a quick figure. Yeah. This is a beta plated sheet right here, and this is an alpha helix. And then also turns is sometimes like a third one, like right here. And so I selected ones that are mostly alpha helixes because those typically in literature are more easy to predict than beta plated sheets. And it's just something we could control. I forget the original question, but that's how I selected the data.
So how is the folding considered optimized?
Okay, so that's also interesting. So this graph is like, I think this graph is good in explaining that. So we have the native structure at the minimum, and the decoy structures are not at the minimum. And this red to blue gradient kind of corresponds a decrease in the energy of the protein, which you can measure the energy of a protein in a multitude of ways. Typically, I like to think of it as the native structure is the most thermodynamically favorable of all possible structures. So this is kind of like the search space where you have energies for each of these decoy proteins existing, but the minimum corresponds to what it'll actually most likely fold down into. You have different metrics that can kind of measure this this minimum, or I measured the deviation from this minimum, which was kind of a roundabout way to do it. But there is a function. There's this bioinformatics software called Rosetta, and anyone who's in a aspiring bioinformatician will probably want to learn PyRosetta, which is their Python implementation that scores proteins scores proteins according to various metrics. It's called the Rosetta. It's like the energy of the protein from the Rosetta package, and that's one way to find the energy or find the minimum that corresponds to the most favorable native structure. I'm sorry, this is a lot more bio than I thought we were going to get into. And any other questions?
Do you have any kind of like idea on how to encode the functionality of the amino acids or like the proteins?
Yeah, so Jeff brought up a good point of just having a discriminator. And
so cut or no cut?
Yeah, that would be the when you think of like the simplest case, it's classifying between a yes or a no.
Is that the only functionalities that proteins do? Sorry for the basic question.
No. So I did a research study. I've been continuing a research project for a while. There's this really good class. There's this class of restriction enzymes there's this class of enzymes called restriction enzymes. And there are 1,200 of them. And they perform cuts along DNA. That's basically what they do. It's used for a lot of DNA biotechnology techniques, such as like inserting plasmids or digesting or like denaturing DNA, bacterial transformation. So that's like one group of enzymes that perform cuts. Like we have ion channels, which are proteins that are integral in the membrane and allow things to pass through. So like another kind of discriminator model could be built so that it differentiates between a model that is an ion channel and that's not, right? And then we have other proteins that are like, there's like marker proteins that are on the outside of the membrane. There are proteins that simply like carry messages that are involved in like signal transduction, which is the idea of taking a signal from like the membrane of a cell down to its nucleus. There are a variety of different proteins.
Okay, so basically our goal for now is to just, given that the protein cuts, generate that protein.
Yeah.
Okay.
And so that kind of takes away from us needing how to encode the function. Instead, we're just using a discriminator to discriminate between those two. And what's good, this kind of goes back to what Yip brought up earlier, is that we already have online, we already have a good, we can mine for that data online because we already have the annotations for proteins that exhibit that behavior.
Okay, gotcha.
Yeah, and this would be cool. I'm very excited. But it's still up to the group to like to what degree you want to explore this term. And often in previous terms we had other projects continuing under different themes, right? So fall term was searching architecture sizes. That was kind of the theme. We did 2 papers out of that. Winter term was kind of stock market predictions because the whole GameStop her fiasco. So this one can be a little bioinformatics heavy if people are interested. But machine learning can be applied to a variety of topics, and there's plenty of ties between bioinformatics and other fields. And if anybody has any other ideas for projects we could explore, I'd be interested in hearing them as well, that are or aren't bioinformatics related. But I'm just procrastinating drafting this, aren't I? Yeah. So.
Yeah, I have no idea for projects now, but I think it'll be cool if we make an API or something, like deploying a machine model, something like that.
Okay.
Yeah, because when I worked with some company once I finish a machine learning model, the next step is always deploying. So.
And yeah, I don't think I've ever deployed a network. I've only ever tested it locally, and then that was it. That's actually a good point.
Yeah, it's quite. I think it's important and a bit fun.
Yeah. How independent is that from the model?
Not independent at all.
Yeah, like this. Go ahead.
Go ahead.
Well, as far as I know, there's two main methods of doing it. One way is like using Google's Cloud Run, where you basically make like a Python file of just loading the model and executing something, and you do it. Another way is you kind of like deploy, save the model in a certain format, and then have this thing called a TensorFlow serving server, which basically every time it gets a request, it runs the model and sends back a response. Those are the two main ways I know. So it's a bit different, but quite similar.
Yeah, I mean, this topic seems very rich. So what we can do is we can dig deeper into this topic and split it into 2 and even 3 papers. So one paper could be just a paper on pipelining a protein net. So we could just talk about it and put it on archive. That could be one paper, and the other paper, obviously, the research itself.
Yeah.
I think
is archive. Oh, sorry. Is archive just like a platform just to publish without like the peer reviews?
Yeah, it's just preprints. That's the technical term. So what people usually do is they publish it to get attention, and then they use the same paper to publish to submit to a conference. So while the conference submission is going on, they use just their preprints.
So it's not like a technical publication.
It's not peer-reviewed journal.
Oh, okay. So is it
anybody?
But it's like Wikipedia. Like anyone can edit Wikipedia, but if you think about it, the quality of it is very high. So same with archive. Anyone can publish it to archive, but the standards is pretty high in terms of the research quality. But yeah, you're right. Anyone can publish anything. So you cannot say I published a peer-reviewed journal when you submit to archive.
And so you publish them.
But you can say, here's my research.
It's published, you know.
It is published, you know. I'm banking on that.
Because you can cite it. That's the good thing about it. You can cite it. And peer-reviewed journals do cite archive papers all the time.
That's interesting, actually. Yeah, so I mentioned that the database is through Protein Data Bank. This would be the data set. This would be Protein Data Bank API calls. And I can introduce the framework. That's actually, I wanted to actually go to GitHub here. I have all of my code from the past summer here, protein structure error predict. A little outline, and then everything that encodes a protein is in this file. Many different functions, a lot of coding, but it was really interesting. And something I didn't bring up is the fact that you want to have whenever you like introduce a convolution neural network, which is what I did learning these, you typically don't want to have like static images. What I mean by static is probably a bad word, but if you're trying to classify hands, you don't want to have a hand that's always in the normal position. You want to have like rotated versions of those hands, so that the model can learn not just like one image of the hand. It can learn how the hand looks when it's upside down. So what I did is I rotated the protein and encoded it in various ways, as a way to allow the model to learn not the absolute locations, but relative locations as well.
I think GANs are pretty good at identifying like structures like that. Like I think like one of the things they're best at is like identifying features that are independent of like rotation or scale or any of those.
I was not sure. So this is not a very well developed plan. Does anybody have any other thoughts? I'm probably leaving out a bunch of things I just haven't thought of.
Well, it's just another question, but for the protein, how is the positions initially encoded? Like, where's the origin of where's the 000 of the protein?
So the 000 is at the, it's in the center, I think.
Oh,
Well,
really?
it's just
I think it's in the
arbitrary,
centroid.
right?
It might be arbitrary.
You could pick any. It's just a reference, right?
Well, when I did the program here, I always had to move it to the centroid. I had a function that was like get centroid. I know I did that. So maybe it's right. The origin is arbitrary, but well, the origin would be arbitrary because these structures are obtained using crystallography in most cases. So it would be arbitrary, and then you'd effectively move the positions to like the center of the protein, and then you'd rotate about its center. That's what I would do. That's what I did.
Yeah,
your
probably.
standard. Sorry.
I was gonna say probably you're not interested in actual like absolute coordinates, but you're more interested in where it stands with respect to other coordinates that you might have. So maybe the distance is more important, and we can even maybe include distance as a artificial feature, but I'm not sure. Probably just the XYZ coordinate is enough.
Yeah, maybe for now I just want to check out Ethan's encoding.
Yeah, no, all these questions, like I appreciate all these questions because someone coming from a biology background definitely takes this for granted. I'll write this explore.
Also, for 3D coordinates, when you have rotation, it is easier to work on polar coordinates rather than XYZ. Maybe
really,
because and then instead of doing XYZ, you can just have the rotation angle and the z-axis and the distance from the center. So, and that makes it easy to manipulate. Did you work on that, or did you just do XYZ?
I used linear algebra to rotate it. I applied a rotation matrix.
Oh, so you worked on X. Okay.
Yeah,
it worked.
Okay. I remember like a while ago when I was working with 3D coordinates. When you do rotation, like, did the order of rotation matter? Like, first, if you rotate around the x-axis and y-axis is different when you rotate around the y-axis and x-axis. Something like that.
I don't think so. I think the axes are independent from one another.
Oh, okay.
As long as like the sequence of rotation matrices that you're using, if you're doing
it's constant.
It's gonna be like the distributive property or whatever, or associative property.
Matrices, Yeah.
I think to rotate in XYZ, you have to multiply with a linear combination of cos or sine. But to rotate in polar, you just add the rotation angle.
Okay.
So I think that's just easier to work.
Yeah. We could add that as a functionality. Oh, this is interesting. Oh man.
Sure, that's like a well-documented problem too. So we could probably,
Yeah, it's probably on Stack Overflow somewhere.
Yeah. We should cite Stack Overflow in our paper.
It's true. I don't know if you're supposed to do that actually. I don't know. Cite. I don't know. That's funny.
Cool. Citing the triangle inequality or something.
Yeah. So yeah, any other questions? Like, I wouldn't be afraid to ask anything that is biology based with proteins, structure. Jeff will go into more in depth about GANs, their general adversarial networks, which implement a discriminator and a generator that kind of compete to form predictions.
Yeah, it's sort of like a game theory.
Yeah,
there's like two players, and then
is it a zero? What's the term for the game?
Zero sum game.
Yeah.
I would need to review.
I know. I'm reading a book by it's not by Nash. It's not by John Nash. It's about John Nash. Beautiful Mind. Well, I've been reading it for the past 5 months. You know how those go. But it's really interesting to think about game theory. So that's why I asked.
Yeah, are you taking optimization theory?
I actually might have that in my plan of study. I thought that was cool.
You'll probably like it because that's the sort of stuff you guys are going to talk about.
Yeah, cool. Thanks. So we're coming up at the hour mark. If you don't really have any other questions, we're just about done here. The next meeting will be on Monday next week, either 6 or 7. I'll relook at the schedules and see what the best time is for everybody, and we can start to get to work, do some coding, and next week will be presentations. Getting ahead of myself.
Yeah, in the meantime, can you check that you can see this repo? Because I know if you just joined, you might need to be invited as a member, not an outside collaborator.
I think this is a private repo.
I can see it, for example, even though it's private, because I think I'm the owner, but I'm not sure if others can see it. Also, we do have virtual enrollment fair next Wednesday. So that will be for promoting the club if you just want to sit and host that, like with me or anyone else. Or actually, if anyone wants to host it, please let me know.
When is that happening?
It's from 4 to 6. It's a virtual enrollment fair next Wednesday.
Next Wednesday.
So, like all the clubs at Drexel will be there, so you can just talk to anyone that you might. So one of us has to host our Zoom link. So even though I registered it, I don't need to host it. Anyone can host it. So let me know if you're interested. And also, if you're interested, please submit an interest form for Drexel AI hoodie. And other than that, I think that's all from my side.
Oh, you found generating models. Okay.
Yeah,
I just saw you sent this.
This looks kind of like relevant. I didn't.
Okay.
I haven't read it, obviously.
Okay. Where do I go to make this public?
You would go to
manage
options, I think, and scroll down.
change visibility.
Yeah.
Oh wow, you guys are just awesome. Type Drexel AI protein. Okay, cool. Public, so everyone can see it. And yeah, now it should be public. And I'm gonna add that resource down here.
Perfect. Also, we kind of assume that everyone knows Git, but if you don't, then we can have a session on this as well.
Okay. Cool. Awesome.
Somebody we could. We don't have to do that next meeting. I guess we could do it like some other meeting. I guess.
Yeah.
Yeah, I can do a small demo.
I would appreciate that.
Yeah.
Still got to catch up on it too, Cool.
That will be probably after next meeting.
Yeah.
All right, sounds good.
Thanks, it was fun. I'm not sure if I'll be in the next meeting because of class, but hope you all have a fun time.
I'll see you, Simon.
We can also kind of alternate, or I'm not sure alternation, but
Oh, okay.
We'll figure out a system.
Yeah.
I'll resend out a poll because now we kind of figure out who's gonna come and who's interested or not. So we can just reevaluate.
Yeah, we can also like set up maybe separate meeting schedules depending on the paper you're working on. Like,
Yeah.
Okay.
Awesome. Well, I'll stick back if anyone wants to hang out, but we're done here.
See ya.
You're
Have a
free to
nice
go.
day.